We're going to take a look at cell size and the glycocalyx in bacteria. As we talked about before, most bacteria are much smaller than eukaryotic cells. So in the picture on the right, we have a red blood cell up here on the top, just part of it because it's so large, can't even fit on the slide. And then below that, we have an E. coli and Streptococcus bacterial cells, and they're much smaller than the eukaryotic cell. And we also looked at a picture that showed that rods or bacilli, such as the E. coli, are generally larger than the cocci, which we have a Streptococcus here. And then just for size comparison, you can see we've got some viruses here which are much smaller than the bacterial cells. They have found a few very large bacterial cells. So in this picture on the bottom left, the smaller cells are actually eukaryotic cells. These are paramecium, which are protists, and they are protozoans, so they're animal-like, they ingest their food. And then here is a species of bacteria that they found living in the intestines of surgeonfish, which live in the waters in Hawaii. And that is a huge bacterial cell. But that's the exception to the rule. Most of the bacterial cells would be much smaller than the eukaryotic cells. And then I know that you've learned this before, why cells are so small, but it's important to remember this. So cells are going to be small because you need to be able to diffuse enough food and waste products across the cell membrane in order to keep the cell alive. So you want a much higher surface area, so the cell membrane size on the outside here, you want that to be much larger than the volume that's inside the cell. So if you take the surface area and divide that by the volume, you can see that this smaller cell up here on the top has a surface area to volume ratio that's higher at 3 than this bottom cell, which is larger, but it has a smaller surface area to volume ratio. It's down here at 1.5. So what happens when you calculate the surface area to volume ratio? The surface area is a squared number, and volume is a cubed number. So as the cell gets bigger, because volume is cubed versus surface area, which is only squared, the volume is going to increase a lot faster than the surface area will. And that's going to create a problem because if your volume is too large, you've got too much stuff inside the cell, and you're not going to have enough surface area to transport all the things in and out of the cell that you need to. So kind of the way that I think about the transport is if you had a city with roads going in and out, okay, and that's how you're going to be transporting food in, waste out, and then if that city grew and the surface area is not going to increase as fast as the volume, so you're going to have a lot more people living inside the city, but your roads, your surface area, hasn't really increased as much, and so you haven't built a lot of new roads. So it's going to be harder to get enough nutrients and waste products in and out of the cell to keep the larger volume inside the cell alive. So that's why the cells are going to be small, because they have a much higher surface area compared to their volume. So you want a high surface area to volume ratio. So the reason cells are so small is because they have to have a large enough surface area to keep up with transporting nutrients and waste in and out of the cell. So now we're going to look at the parts of a bacterial cell, and we're going to start on the very outside. So we've got the cell wall, which is in brown here, and we're going to be taking a look at the things that are outside the cell wall first. And so we'll start with the glycocalyx. These are the structures that are found outside of the cell wall in a bacterial cell, and bacteria do not have all of these. So they may have some of these, but a lot of times they're not going to have all of these. So it depends on the species which of these they will have outside their cell wall. So we're going to focus on the glycocalyx here, and then we'll talk about the flagella, axial filaments, fimbriae, and pili later. So the word glycocalyx, glyco refers to sugars, and calyx means covering. So the glycocalyx is a sugar coating on the outside of the cell wall of these bacterial cells. And so it's going to be made out of polysaccharides. And we have three different forms that we can see with this sugar covering, this glycocalyx. So some bacteria have capsules, some have slime layers, and then many bacteria can form biofilms, and that's actually part of their glycocalyx, and it's called extracellular polymeric substance, or EPS. So the functions of the glycocalyx, it's definitely there for protection. So for example, the capsule on some of the bacteria makes them slippery and hard for some of our white blood cells to phagocytize them because they just slip away out of their grasp. And then also with protection, this glycocalyx will help prevent the bacterial cells from drying out. And then the other function is attachment. So the glycocalyx is going to help the bacteria stick to surfaces. And that's especially true with the biofilms, and we talked about that sticking to your teeth, and that allows the bacteria to stick there, and you've got to scrub them off with your toothbrush. So the capsules are very thick, they're well organized, and they're going to be hard to remove from the cell. And capsules are found on some of the bacteria, not all. And the ones that do have capsules, they'll use the capsules to stick to surfaces, and it also serves as protection. So it makes them slippery, makes it so some of our white blood cells can't phagocytize them, and then also protects them from drying out. Some bacteria have a slime layer, and the slime layer is different from the capsule. It's still made out of polysaccharides, but it's much thinner. It's not as well organized, and it's easy to remove it from the bacterial cell. It also serves the same function as the capsule, so it's there for protection, helps the bacteria stick to things, and because it's slimy and slippery, it also aids in motility and helps the bacteria move around. And then the third part of the glycocalyx that we find with some bacteria is the biofilm. And that's called the extracellular polymeric substance, or EPS. And that just helps the bacteria stick to each other and to surfaces. And then also it's there for protection as well, because it's hard for antibiotics to penetrate that, and also the white blood cells. And this is just a picture, some drawings of the capsule. Capsules are going to be much thicker than we find with the slime layers. Slime layers are going to be much thinner, and the capsules are harder to remove than the slime layer. And then the biofilm is just that slimy stuff that the bacteria will secrete, made out of the polysaccharides that help them attach to surfaces. Here are some pictures of some bacteria that do have capsules. So at the bottom we have Streptococcus pneumoniae. That does cause pneumonia as well as some other diseases, which we'll talk about later. And you can see in yellow the thick capsule that's surrounding these bacterial cells. So that's all made out of polysaccharides, and it's there for protection and helps the bacteria stick to surfaces. And then just as kind of a review, the shape of these bacterial cells. So we've actually got two bacterial cells that are stuck together here, and so this would be a diplococcus, because you've got two cocci that are stuck together. And then up here on the top we have another bacteria, Pseudomonas aeruginosa. We'll talk about that one in the next chapter as well. And this one, they've done a negative stain to be able to see the capsule. So they've stained the background, and then you can see this clear area, whoops, that's around the bacterial cell. That's the capsule, okay, made out of those polysaccharides. And that helps the Pseudomonas aeruginosa bacteria, helps it to attach to things, and also is there for protection.